Fantastic. Well, thank you guys so much for having me today. And I'm so sorry I'm not able to be there in person. I have clinic today, so I'm in my scrubs and I'm ready to go as soon as this meeting is over. Thank you, Chris, for the introduction. As you know, my name is Sofia Vignolo. I'm an MD PhD student at Oregon Health and Science University, and I'm really excited to share just a portion of my dissertation work that's focusing on engineering scaffolds that mimic biology for enhanced bone regeneration and craniofacial applications. And I wanted to put this a little bit more in the context of faith-based contributions. Um, Again, as I mentioned, I was really honored to be able to be invited to speak at this forum. And my work is focusing on this data set that includes rats. It's a first data set for FaceBase that has rats. It's a pretty large data set. I just put everything that I had on there. So it's about 150 gigs, over 240 files that has longitudinal in vivo micro CT data, as well as ex vivo high resolution micro CT data, and lots of histology. So I will dive right in the sake of time. To give some context and clinical background, the motivation for this is to treat bone defects, and bone defects can arise from a variety of causes. Hopefully, you guys can see my cursor over here, from cancer resection to birth defects or infection. And these large defects simply don't regenerate on their own. We term them critical size defects, and they still remain one of the most challenging clinical problems in reconstructive surgery. And autografts, which are bone grafts taken from the patient's own bodies, are still the clinical gold standard for treating these defects because the patient-derived bone has like that perfect mechanical and mineral cellular cues that are needed for healing. But autografts are limited in supply, as we know, since taking bone from one site creates an injury in another, and that can lead to donor site pain and complications. And so engineered grafts have made lots of progress, but they still struggle to fully replicate that complexity of native bone. And as a result, the existing clinical options really lack that level of biomimicry that's needed for reliable functional bone regeneration. And typically, that bone graft fabrication pipeline starts with patient imaging to assess the defect and design a personalized approach. And then you can use different biomaterial strategies like cells, bioactive factors, or scaffold material that are combined with fabrications like 3D printing or other ones to create this implantable construct that's personalized for that patient. And in terms of the bone formation pipeline, just to understand what's happening at a cellular level, bone formation progresses through coordinated stages that are regulated by cell matrix interactions. Physiologically, craniofacial bones begins when stem cells start to proliferate and gather into condensations. These condensations then become an ossification center where RUNX2 is upregulated, and they drive these cells to differentiate into osteoblasts. The early osteoblasts start to secrete collagen as vascularization infiltrates to deliver nutrients, cells, and other chemical signals, or all the goodies. And then the mineralization process begins, and these osteoblasts become trapped or embedded within that matrix, and they start to mature into osteocytes. And then over time, you have that continued remodeling that transforms that very early tissue into the beautiful organized bone. And in terms of the materials of bone, collagen and mineral are the two major structural components. And so being able to modulate these elements in a scaffold is what allows us to recreate these stages. So in my work, I use collagen density and mineralization as tools to represent these distinct phases of bone formation. I have soft collagen to represent that early stem cell recruitment and permissive cell migration. I have high density collagen that mimics matrix compaction during early osteoid formation, and partially mineralized gels to capture the onset of calcification, and finally, fully mineralized gels to represent the mature bone matrix. And together, these engineering approaches really allow me to recreate this timeline of bone formation in a controlled and modular way. And like I mentioned, more starting with that soft and permissive matrices and progressing towards more dense and mineralized environments that mirror the later stages of bone regeneration. And matrix mineralization is a defining feature of the bone extracellular matrix, and it's influenced by, or it influences stiffness, nanotopography, ligand exposure, and osteogenic signaling. Prior work from our group has shown that the mineralized collagen fibrils appear really smooth, as you can see here, and loosely arranged, whereas the mineralized fibrils become densely packed with that interfibrillar mineral. And together, these challenges directly influence how cells are able to interpret their environment. So, if we're able to tune or change that mineralization, we can really modulate mechanosensing, stem cell fate decisions, and ultimately osteogenic signaling. So, it makes mineralization a really powerful and physiologically relevant design parameter. And so, the work that I'm presenting here today really sought to isolate the specific role of mineral content in early osteogenic priming and in vivo bone regeneration. And we wanted to understand, specifically for this paper, independent of the density of the matrix, how much does the mineral itself drive osteogenic commitment and downstream bone healing? So our hypothesis was that if we increase that mineralization, enhanced, we're going to enhance this process and be able to have accelerated osteogenesis. And so to give you a little bit of an overview of how we did this, we were able to fabricate the collagen hydrogels and then densify them using centrifugation. And collagens are like largely made of water. And so this is like compressing a fibrillar network. So like squeezing water out of a sponge. And then we finished the cross-linking so that it remains compact. We then introduced a biomimetic mineralization step that allowed us to generate constructs that represent those distinct physiological stages of bone formation that I mentioned earlier. So I wanted to dive in a little bit about this mineralization method because it's pretty unique to our lab, and well, some other labs also use it now. um, to mineralize our constructs, we expose them, the high density collagen, to biomimetic mineralization media, and this contains calcium and phosphate ions as well as osteopontin. Osteopontin is especially important here because that native bone usually secretes, or the osteoblasts in the native bone secretes osteopontin during that early matrix deposition and is able to control how the mineral forms inside of the collagen. So physiologically, osteopontin is able to delay nucleation of these calcium and phosphate ions enough time to stabilize and create this amorphous calcium phosphate precursor. And that allows minerals to be able to penetrate inside the collagen fibril, leading to intrafibrillar mineralization. And we supply osteopontin in the media to recreate this native process rather than letting the minerals just passively deposit on the surface. So this means that we're not just coating the surface with minerals, but we're really trying to recreate that hierarchical organization of bone at the nanoscale. And as a result, we can generate constructs with tunable degrees of mineralization depending on the time that it's exposed to the media. And to verify that our mineralization method produced these distinct matrices, we performed structural, compositional, and mechanical characterization. We were able to confirm the microgel size and morphology across all the groups. We saw that expected nanoscale compaction with increasing mineral deposition, and alizarin red here on the bottom specifically binds to calcium. So as mineralization increases, that calcium deposition also increases, seen in this deeper red staining, and also the opacity increases because matrix becomes less light permeable. So you'll see those images getting darker as it's more mineralized. And FTIR also confirmed this quantitatively with higher phosphate peak intensities, confirming that there's that increase to mineral matrix ratio in the partially and fully mineralized gels, respectively. And mechanical testing also showed that corresponding increase in stiffness. So together, overall, this data shows us that we can reliably and incrementally tune mineral content, nanotopography, and stiffness using our controlled mineralization process. And to test how mineral content affects osteogenic signaling, we examined nuclear RUNX2 in human stem cells that were seated on the surface, there we go, of these hydrogels. Interestingly, we saw this greater osteogenic commitment in the fully mineralized gels, which is reflected in the increased RUNX2 within the nucleus, as you can see here stained in green. And these fully mineralized gels showed greater percentage of RUNX2 nuclei at 24 hours, and this trend persisted at the gene level at 3 days. And this is pretty fast, considering that standard in vitro cultures using osteoinductive media, usually RUNX2 activation takes several days to a week. So the fact that we're able to see this kind of level of commitment just at 24 hours suggests that maybe providing a physiologically relevant mineralized environment can accelerate osteogenic programming without the need of osteoinductive media or these other bioactive factors. And then the fun part, because I know you guys all love craniofacial anatomy, we implanted the microgels into rat critical size Calvarial defects, and we were able to compare the mineralization dependent effects on bone healing. And so this consisted of creating an 8 mm bone defect in the skull. And then we implanted these microgels and monitored that regeneration over time. We were able to have longitudinal scans that I'm not showing today in today's presentation, but it is on FaceBase. And we did that at 4 weeks, 8 weeks, and 12 weeks. And then we had a final, more higher resolution ex vivo scan at 12 weeks. And that matches with the histology data as well. So those are the two data sets I'll be showing today. So the micro CT at 12 weeks post-implantation revealed some striking sex-dependent effects. And we heard this a little bit earlier, so it's important to keep this in mind. So in females, microgels were able to produce increased bone formation compared to the empty defect, as you can see here. But none of the gel groups here approached the autologous bone benchmark, and the effects remained largely unbridged. And then in males, fully mineralized gels did enhance bone formation compared to the non-mineralized groups and empty defects. And we saw this more spatially continuous regeneration extending from the defect margins. And these differences that we see, these sex-specific differences, align with known sex-specific patterns in growth. So this is the growth curve of this specific rat strain. And it's aligning with these patterns that we know that are happening in growth, metabolism, and just the baseline bone turnover over the course of bone healing. And overall, mineralization here acted as a pro-osteogenic cue in males, but it had a limited benefit in females. So this really emphasizes the need for personalized sex-specific considerations in scaffold design. And histology at 12 weeks post-implantation shows a bit of a deeper look at the impact of mineralization on scaffold integration, degradation, and also bone formation patterns. So starting with an empty defect here on the left, we kind of see exactly what we expect for an empty 8 mm critical size defect, which is almost no bone formation in the middle. So here at the top, we're looking at a coronal section, and here's the edge zoomed in, and here's kind of somewhere within the defect. The space within the middle is largely filled with this fibrous connective tissue. And then when we implant the microgels, kind of these groups in the middle, we start to see more bone formation from the periosteal surfaces along the margins, but the amount of bone and the continuity of bone across defects remain variable. And the autologous bone here, all the way on the right, showed us this robust new bone deposition around the implant and having kind of near complete defect closure. What was really interesting is that the new bone formed all around it. You can kind of see here the original graft labeled in G, and then the new bone labeled as NB, just forming all over it. So it kind of just made it even thicker than the original bone. And taking a deeper look at the histology, the fully mineralized group had more residual scaffold material that remained within the defect, and I marked these with asterisks. And that indicates that these dense, really mineral-rich gels likely degrade more slowly. And importantly, even when these residual gels were present, newborn that I marked with NB consistently formed all over around it. So this really shows us that these constructs are supporting the two fundamental behaviors that we want from a scaffold for bone repair, which is that they're osteoconductive, they're acting as this structural scaffolding, and they're osteoinductive because they're promoting that early osteogenic activity around the defect. And so from this work, we have learned that progressive mineralization increases stiffness, nanotopography, and calcium phosphate content. Mineralization alone enhances early mechanotransduction and osteogenic priming without the use of biochemical supplements. And mineralized microgels really enhance the bone regeneration and shape the spatial distribution of new bone with a sex-dependent healing outcome. And this entire data set is uploaded to FaceBase, and it's currently under embargo until we get that final publication. I showed you here the mineralization story, but there's also a separate density story, and kind of focusing on what's the effect of density in this pipeline. So that's currently still pending. But hopefully we're hopeful to have this out sometime this year. And as soon as that publication is out, then we will let FaceBase know and release it to the public. So we're really excited about that. And as I mentioned, this is probably the first longitudinal rat craniofacial bone regeneration data set on FaceBase that has lots of these multimodal of the in vivo, ex vivo, micro CT, and histology. So that's really exciting. Yeah, so this is just a little of the tidbit of what I added on this paper that's already published to just increase that awareness of that this data is available to be shared and be reanalyzed. And some future directions in terms of where this work is taking us. And these are more like current directions because we're actively collecting data on this. But we've expanded from using acellular to cell-laden gels by modifying the earlier methods that I mentioned and added HUVECs or vasculature cells and stem cells to the collagen solution. We were able to successfully recreate this pericyte-supported vasculature, and they're embedded in a mineralized matrix. And histology also showed that the matrix is much denser and organized due to cellular remodeling when compared to the acellular group. And we established this decellularization method that preserves the collagen architecture, mineralized organization, and vascular channels while removing the immunogenic cellular content. This is currently being evaluated for a patent, so it's all TBD. And we are very excited also to show some preliminary data where these decellularized gels are accelerating bone regeneration on 5 mm defects as early as 6 weeks, which is super exciting. They're looking like the autologous bone by 6 weeks. So I'm like, oh my gosh, this is so much fun. It's also really exciting to see these spatial thermal maps that shows the bone ridging at the center of the defects. And you can kind of see here how these, and I can use, yeah, these early gels start as an isolated island, and throughout time they become integrated with that bone front. And they look pretty seamless. So looks so much fun. And just for the sake of this talk, I wanted to have some more future perspectives of bone tissue engineering. And I wanted to end by highlighting where this work points us to in that broader landscape of this field. One major direction is the development of smart and adapted scaffolds that evolved with the healing process. As we have seen, bone healing is not a static process. It progresses through phases that require distinct microenvironmental cues at different time points. Early on, the cells may need, softness, porosity, and space to infiltrate, and later they may need higher stiffness, mineral content, and pattern architecture to be able to support that differentiation, that new bone formation, and the remodeling process. So next generation scaffolds to try to aim to address this by responding spatially and temporally. And the second opportunity kind of builds on this idea by trying to integrate dynamic cues using computational approaches like AI-guided scaffold design. AI can help perhaps optimize parameters that we've already discussed, like matrix density, mineral content, and even vasculature or vascular architecture, degradation kinetics, etc. And they can use these as inputs for in silico modeling, predictive optimization, and digital twin systems. And so in that way, by creating standardized, high-quality shared data resources like those found in FaceBase, these lessons learned from this work combined with the insights from the broader field can serve as training data for computational systems that are capable of designing scaffolds that are perhaps far more integrative and predictive. And finally, all of this kind of enables that ultimate goal, which is to have personalized clinical care for functional bone regeneration. And so bone graft substitutes could be designed to match the patient's specific geometry, their mechanical needs, and their healing trajectories, combined with a high throughput workflows that bring us closer to therapies that are tailored to each defect for each patient. And with that, I would like to thank lots of people because I showed you only a portion of this work, but it takes a village. But specifically, I wanted to thank my lab, the Bertelsoni Lab, and its members, as well as our funding resources for being able to make this work possible. And I'll happily take any questions you guys may have.